Carbohydrates are a large group of biomolecules ranging from small sugar molecules called monosaccharides to large carbohydrates referred to as polysaccharides. These carbs, as we call them, form the food we eat. They form table sugar, bread, pasta, croissants, hot dog buns. All of the things that we call carbs are foods that are rich in carbohydrates. Honey is a mixture of two monosaccharides, glucose and fructose. Monosaccharides, such as these found in honey, are going to be the main fuels for cellular work. In cells, glucose is used to form ATP. Monosaccharides are also used as the raw material to form other kinds of carbohydrates in the cell. Monosaccharides can be bonded to form more complex sugars, such as 2 to 10 monosaccharides linked to each other, or polysaccharides, which would have hundreds of monosaccharide monomers bonded to each other. Monosaccharides also have the characteristic of having a ratio between the number of carbons, hydrogens, and oxygens. The formula is always a 1 to 2 to 1 ratio, with the number of hydrogens being 2 times the amount of the number of carbons and oxygens. You can pause the video here and actually count the carbons, hydrogens, and oxygens and verify this unique formula for monosaccharides. Another way monosaccharides can vary from each other is in the number of carbons that form these molecules. We have groups based upon the number of carbons. So we have a triose for 3 carbons, a tetrose with 4, pentose with 5, and hexose with 6 carbons. It's the hexose that we'll use most often talking about monosaccharides. In solution, many monosaccharides form rings. In the diagram here, you see common way that a monosaccharide is symbolized. Starting from the far left, we see the structural formula showing all of the carbons, hydrogens, and oxygens. The middle figure has removed most of the carbons, those that are found in the ring itself. It's implied that they are at those indices, those apex points of the hexagon. And in the most simplified structure, all of the structural arrangement is removed and implied by the hexagon structure itself. Along with the number of carbons, the position of hydroxyl groups will also vary the molecules from each other. So here we see glucose, fructose, galactose, deoxyribose, and ribose. Let's take deoxyribose and ribose. These are 2 pentose sugars. Deoxyribose is the sugar that's found in the structure of DNA. It's why we call it deoxyribonucleic acid. Ribose is the sugar found in RNA, and we call it ribonucleic acid. And the difference between the 2 sugars is the presence of that hydroxyl group. 2 monosaccharides can bond to form a disaccharide. The disaccharide is our second group of carbohydrates. Dehydration synthesis is the reaction that's used by the cell to form the disaccharide molecule. Here we can see glucose and fructose bonding to each other to form sucrose. Other kinds of disaccharides are maltose and lactose. Let's animate that to see the process.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of 2 linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
Here we see two glucose monomers that are going to be joined to each other to form maltose, the plant sugar that was described in the animation. Note that hydrogen and the hydroxyl group are used to form water, and that allows for a bond to be made between the two monomers. This kind of covalent bond between monosaccharide monomers is referred to as a glycosidic bond. The last group of carbohydrates are the polysaccharides. Polysaccharides are polymers of monosaccharides. They can function in the cell as a storage molecule, ready to give off monomers of monosaccharides that can be used as fuel for the cell. Here we see many monosaccharides linked to each other to form this particular polysaccharide called glycogen. So let's animate the process of making a polysaccharide.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Starch, glycogen, and cellulose are all examples of polysaccharides. Let's take a look at these and one more important polysaccharide. Starch is the first. Starch is a polysaccharide composed of glucose monomers and is used by plants for energy storage. Potatoes and other root vegetables form starch. Glycogen is a polysaccharide composed of glucose monomers, but in a different pattern. Notice the branching pattern coming off of the main polysaccharide chain. Glycogen is used by animals for energy storage. We store glycogen in our liver, and the liver cells can break down glycogen, forming glucose. That goes into our blood. Cells will pick up that glucose and use it for energy, like in muscle contraction. Cellulose is a polymer of glucose and forms plant cell walls. Note the three polysaccharide chains that are linked together by hydrogen bonds. This makes for a very fibrous and strong polysaccharide. The last one is chitin. Chitin is a polysaccharide used by insects and crustaceans to build their hard exoskeleton, their outer skeleton. Now that we've gone through the groups of carbohydrates, be sure that you can answer these questions before you leave this video. Okay, ta-ta for now.